In this video, we'll take a closer look at protein structure and function. A common thread that you'll come across when talking about organic macromolecules is that function of these macromolecules follows from their structure. Proteins can serve diverse functions inside the cell because they have diversity in size and shape, as well as chemical properties of their amino acids. Proteins fold in three-dimensional space to form organized structures that have a specific function inside cells. A few examples of such proteins are shown in this image. So, how do proteins look like they do? All proteins have just four basic levels of structure: primary, secondary, tertiary, and quaternary. We'll look at each one of these levels in detail. The primary structure is the amino acid sequence that makes up a protein. To recognize the importance of this amino acid sequence, let's look at part of the amino acid sequence for the protein hemoglobin, which is present in red blood cells. The normal amino acid sequence of this protein contains glutamate in the 6th position. If that amino acid changes to valine, the protein cannot maintain its shape, and thus the red blood cells lose their natural shape. This altered form of hemoglobin cannot bind oxygen very well, leading to a condition called sickle cell anemia. So sometimes even a small change in the primary structure of a protein can have big consequences on its function. Let's look at the primary structure of a small protein called ubiquitin. Ubiquitin is responsible for marking other proteins for degradation. The primary structure of ubiquitin is a sequence of amino acids as shown in this image. Hydrogen bonds are involved in the formation of secondary structure in a protein. These hydrogen bonds occur between the carbonyl group of one amino acid and the amino group of another. Hydrogen bonding between sections of the same backbone is possible only when a polypeptide bends in a way that puts the carboxyl and the amino groups close together, forming structures known as alpha helices or beta-pleated sheets. The secondary structure of a polypeptide depends on its primary structure because the sequence of amino acids determines the way in which they interact with each other to form the secondary structure. Some amino acids are more likely to be involved in the formation of alpha helices, others are more likely to be involved in the formation of beta-pleated sheets. Now that we've looked at the primary structure of proteins, let's look at ways in which amino acids within the primary sequence interact with each other to form secondary structures. Here's an example of a protein. Both the primary sequence as well as the folded structure are seen on this page. One of the elements of secondary structure is the alpha helix, formed by the amino acids highlighted in green. The alpha helix contains amino acids that form peptide bonds with each other. Both the peptide bond and the carbon-to-carbon bond are flexible and can rotate, allowing the amino acids to flex and bend in order for their side chains to interact with each other. Amino acids in nearby regions can hydrogen bond with each other to form a secondary structure known as the alpha helix. This structure is commonly found within most proteins. These hydrogen bonds stabilize the structure. This is a 3D ribbon model of an alpha helix. Another common secondary structure found in proteins is the beta sheet, formed in this case by the amino acids highlighted in yellow. On looking at these sheets closely, you can see that there are hydrogen bonds forming between amino acids that are further away from each other. This leads to the formation of strands. Again, hydrogen bonds stabilize these structures. And here's a 3D ribbon model of a beta-pleated sheet. Note that the two strands of the sheet are anti-parallel to each other, as the amino acids wind in a U. The next level of protein structure is the tertiary level. The tertiary structure of a protein results from interactions between R groups or between the R groups and the peptide backbone. These connections cause the backbone to bend and fold, in turn contributing to the distinctive three-dimensional shape of the polypeptide. These R-group interactions include hydrogen bonds, hydrophobic interactions, van der Waals forces, which are weak attractions between various functional groups, covalent disulfide bonds, and ionic bonds. This image shows the various interactions that can take place. There can be hydrogen bonding between the side chains of amino acids that are present in the vicinity of each other. There can be hydrophobic interactions between non-polar side chains of amino acids. Disulfide bonds can form between sulfhydryl functional groups. Amino acids on different strands can interact via ionic bonds. All of these interactions lead to the great diversity in the tertiary level of protein structure. For example, you can get proteins that consist mainly of alpha helices, or ones that are composed mostly of beta-pleated sheets, and others that are rich in disulfide bonds. The final level of protein structure is the quaternary level, where many distinct polypeptide subunits come together to form a single structure or complex having a particular function. The interaction between two or more distinct polypeptide subunits produces the quaternary structure. As shown in panel A and B in this figure, you can see that for the Cro protein, two subunits interact with each other to form a dimer, whereas in a hemoglobin molecule, four subunits interact to form a tetramer. The four subunits of hemoglobin come together to function as one complex. What roles do proteins play inside the cell? We're all made up mostly of proteins. Proteins inside the cell play a crucial role in catalysis. Many enzymes are proteins and they speed up chemical reactions that take place inside cells. Proteins are also involved in defense in the body. For example, antibodies and complement proteins attack pathogens that invade cells. They're also involved in movement. There are several motor and contractile proteins that enable the cell to move, or for molecules inside the cell to move from one location to another. Proteins are involved in signaling between cells. They provide rigidity and structure in addition to functioning as carrier or transport proteins, shuttling materials from one part of the cell to the other. Several membrane proteins regulate the movement of ions and molecules across cell membranes. This concludes our video on protein structure and function. At the end of this section, you should be able to answer questions that are listed on this slide. Thank you.